think this is recording. So, the discussion session for today is about this cool paper that came out just last month. Systematic discovery of antiphage defense systems in the microbial pan genome. And here's a picture of E. coli with a bunch of, I don't know if that's T4 or lambda, I don't think it's close enough to tell the difference. Very cool stuff. And so this is what they did. Here's the paper. It's a science paper. They discovered candidate gene defense systems. And so rather than looking for specific kinds of defense genes, they developed a scheme to look for any genes that might be involved in defense. And they did this, so, and then based on the fact that they co-localize with other defense genes. So it turns out that defense genes tend to be linked together. And so, for example, you guys, I hope, are familiar with restriction modification systems in bacteria. These are enzymes that modify DNA by methylations or that sort of thing. And if that modification isn't present in DNA, they cleave it. And so there's three proteins that are associated with these restriction enzymes. The restriction enzyme itself, the DNA cleaver, the methylase, and then the sequence recognition protein. Those three genes are always found in an operon. And so they constitute a system. They assumed that this would be true for other kinds of defense system genes. And it turns out, in fact, that defense system genes tend not just to be linked together, but multiple defense systems. The genes tend to be found in clusters and islands. And so they presumed that this would be true of things that they didn't know about yet. And so what they did was they looked for genes that were specifically linked to known defense genes and were linked together with each other and tested them to see if they were actually defense genes. And so they identified candidates. They showed that some of them would actually confer resistance to phage or plasmid by cloning them into E. coli or Bacillus subtilis, these gene clusters, and seeing if those clones were now resistant to infection. I'm going to go through each of these in just a moment. And then they would knock out one of the genes in the cluster and show that they're no longer resistant. And in some cases, they could propose mechanisms for how they work, because although they didn't know what the genes were specifically, there were domains within the genes that had functions that gave clues to what they were. So let's be specific. And these are from the paper itself. And so what they did is they went through known defense genes in all the bacterial and archaeal genomes and identified all of the known defense genes. So what are these? These are restriction modification enzymes. Type 1, type 2, type 3, type 4. There's several different kinds, right? The type 2s are the ones we use in the lab for the most part. And CRISPR genes. And then they cataloged all the genes in each genome that are within 10 open reading frames of one of these known defense genes. They considered 10 open reading frames to be close in either direction. And so they made a catalog of all the open reading frames for each genome that were found within 10 open reading frames of a known defense gene. And then they compared all the genomes to see which of these open reading frames occurred near a known defense gene more than once, or which ones occurred most frequently next to them. And identified those open reading frames that were most commonly found near a known defense gene, and called these the keynote genes. And so here's a protein family. So you guys know what PFAMs are, protein families? These are clearly homologous genes that are found on a wide variety of organisms. They're called PFAMs, protein families, homologous genes. And then once they had identified these open reading frames that were clearly associated with known defense genes, and this is a plot of this, fraction of members occurring within gene proximity versus, so for an entire genome, what fraction of the open reading frames are found within 10 open reading frames of a known defense gene? Most of them are not. And then here are P families, and it's these are way over here that they're looking for. These are known defense genes which are clearly clustered to each other. And so they're looking at these open reading frames. Once they've got key open reading frames identified, they ask themselves, okay, what open reading frames are associated with these open reading frames specifically? Because they're assuming that if it's a defense system, that the open reading frames are going to be linked to each other, either as an operon or something almost like an operon. That's what they're doing here. And then whatever the usual arrangement of those open reading frames in, they call that a system, a gene system. They did 2 passes with this, and so these are the genes that they find. Well, 30% of their hits, 102 open reading frames, were to other known defense systems. And so if you start with one known defense system, you find other defense systems are clustered, co-localized in the genome with them. They find a bunch of things that are essentially genes for transposons, right? These defense genes are associated with transposons. And so you also find just generic transposon genes, things like DNA cleavage enzymes associated with mobility of DNA, transposases of various kinds, etc. Antibiotic resistance genes are in this category right here, which are associated with these, which are associated with these, and so you pick those up. You find some genes that are just occasionally associated with them, and they don't know what those are, but these ones they considered candidates. And so these were clusters of genes that are strongly associated with known defense genes and with each other. They ran a second prediction cycle with just these, and they find, again, some of these turn out to actually look like transposon genes. Some more that turned out on closer scrutiny to include known defense genes. These are primarily CRISPR things, Cas proteins. And then narrowed it down to 22 clusters of genes that they found were strongly associated with known defense systems in terms of their genetic organization and with each other. All right, so you've got these candidate operons, and you think that they may have something to do with host defense because they're clustered with other host defense genes in a wide range of organisms. So you clone them and you test them, right? And so they've got donor organisms. For each one of these 22 gene clusters, they identified the best donor system. And so what are these? How do they identify them? Well, one is that the genes ought to be in the consensus organization. They're going to clone each of these into both Bacillus subtilis and E. coli, and so picking a close relative of Bacillus subtilis and E. coli is a good idea. So you're cloning from Bacillus licheniformis into Bacillus subtilis instead of something way off the beaten track, some cyanobacterium where that gene may not be expressed very well in E. coli or Bacillus subtilis. Notice that you also have to choose defense systems that aren't already present in these organisms, so that these strains here were specifically chosen because they don't have any of the defense systems that they identify as candidates. Some E. coli strains have some of them, some Bacillus subtilis strains have some of them, but they pick strains of these two that lack all of these. They are variable from strain to strain. They pick a donor system. They didn't clone these things. They just synthesize the whole genes. Clone them into E. coli and Bacillus subtilis, and then they challenge them. Now, why do it in both Bacillus and E. coli? because these have very different phage that infect them. The gram-positive phage are quite different, generally speaking, from traditional bacteriophage. And so they challenged the Bacillus subtilis strain with a bunch of Bacillus phage and the E. coli one with a bunch of E. coli phage and see what happens. And so this is the graph. They've got, this particular one is the Bacillus subtilis set of infections now. And they've got each of these systems, which they've given these names from Greek gods and stuff like that. Here are the donors. So here's a system of Thoris. They cloned one from Bacillus amyloliquefaciens, one from Bacillus cereus. They cloned that into Bacillus subtilis. They challenge it with SPO1, which is a typical bacteriophage in Bacillus. And this one provides 10,000-fold resistance to phage. In other words, to get the same number of plaques on a plate, you have to have 10,000 times as many phage in your initial infection. That's pretty good protection, right? And so each one of these reds represents some level of resistance to infection compared to the Bacillus strain without this system cloned into it. And this is showing the clusters. So SPO1, SPS5J, 5C, 5-3T, SP beta, and so forth and so on. Some of these are more resistant than others. Oh, and as a positive control, they cloned in this disarm, which is it's neither a CRISPR nor a restriction system. It's a different system of Bacillus. And then two different restriction modification gene clusters. Notice that none of these work as generally well as restriction. Restriction enzymes are the primary host defense in bacteria. And CRISPR is probably the next one. Disarm is good in Bacillus. And so these aren't generally as effective. This one's not bad. But they are effective. All of these had some effect on some of them. Of course, these are the ones they're showing because they put 22 in, and these are the ones that came up positive, right? This one wasn't so good. And so all of these confer at least 10-fold resistance to bacteriophage, and sometimes, up to 10,000-fold resistance. And here you're showing that the importance of this thing is that it's showing you some of the genes and what they might include. So this one includes a helicase. What on earth good would a helicase do for a host defense system? Well, these viruses inject DNA, right? And so oftentimes the first step to doing something to the DNA is to pull it apart so you can make modifications or cleavages or whatevers. There's a gyrase in one. Here's an endonuclease. That's pretty obvious, right? If you could recognize the DNA as foreign, then you can break it. And that's what, so this one, you're beginning to see what they might do. ATPases, there's a wide range of things you can do with ATPases, but again, prying DNA apart, providing the energy to provide DNA apart is one of them. Here's the one for E. coli. So they've got 4 different types here. These are more complicated systems generally. And then these different viruses, T4, T7, lambda, etc. And you can see that they all confer some resistance in one form or another to some of these phage. Notice that they're pretty specific, right? Some of they work against some viruses and not others. In some cases, this might do with how the DNA is introduced into the cell, what form it's in. So 527, for example, injects single-strand DNA. T7 injects double-strand DNA. So there might be specific systems depending on how they're infected. Some of these have transmembrane proteins. We'll get to that in just a minute. There's an interesting caveat to this, which is also true for restriction modification systems. They quantitate this based on the number of plaques. but the plaques you get look exactly the same as normal plaques. This is true for restriction enzymes. And so what you're really looking at is that very first infection, the first virus that gets into the cell. Because once that virus has been successful in infecting a cell and producing offspring from that, those offspring are completely resistant to the antiphage activity of these things. Like a restriction enzyme. Remember, if the restriction enzyme, if the virus gets in and somehow avoids being broken, the newly synthesized DNA will be modified and will no longer be recognized as foreign by the restriction endonuclease. And so something along those same lines must be happening here, because the plaques you get are the same size, there's just not as many of them. So what are these things? So they go through several of these. Of course, the ones that work the best and the ones they have the best data for. One of them they call Zoria. Type 1, which contains these four open reading frames, and type 2 that contains these 3. They've got examples from Burkholderia, Desulfovibrio, Pheromonas, Sanguibacter. And you can see they're clustered with known defense genes, this type 1 restriction modification thing, and this other, it's a disarm called Wadjet. Here's a transposon, here's another restriction modification. These things really are clustered. Let me see. They typically are in traditional operons. It doesn't show it here, but there's actually a terminator, and this thing has its own promoter here. So these aren't in the same operon, even though they go the same direction. This is a discrete operon. There's a couple of other open reading frames that are associated with this, but they're not consistently associated with that. Let's see. This is another one, Zoria. It's got 3 open reading frames. It's associated with it. Associated with one of the other ones that they just identified. And type 1 and type 4 restriction systems. Here's an integrase, so that's part of a transposon. This one is associated with these other restriction enzymes. So these things really are tightly clustered, and that's no surprise, right? That's how they identified them. Here's the Zoria system again. It's got these 3 or 4 proteins. They divided them into 2 classes because all of them have these 2 open reading frames, and then they either have these 2 or this one. This one's an endonuclease. That makes sense, right? You're going to cleave it. These ones are not. These are helicases, and all of these are vaguely homologous to proton channels. So these things are homologous to the proton channel part of the flagellar motor. Now, what on earth good does that do? Well, they haven't tested it. They don't know for sure. But their hypothesis is that if these things are recognized or if they're activated by the presence of a virus, and they have no information about how that does it, if these things become active, the presence of just those proteins would open a channel in the inner membrane and depolarize the membrane. If it's an open proton channel, all of a sudden your electromotive force goes to 0. You've got no proton gradient, which is lethal. And so their hypothesis is that the way this thing works, at least this one, well, either of these, is that bacteria that are infected commit suicide. Now, that's pretty unusual for bacteria. It's quite common in multicellular creatures, right? That's the primary defense that you use in your body is apoptosis. You got an infected cell, it commits suicide, or it's killed by a macrophage. But here, their hypothesis is that you have in a bacterial population, the first cells that get infected commit suicide by membrane depolarization. Now, I will argue that that's probably not as simple as that, because the question is, what is this helicase for then? Or what is this endonuclease? And their counter-argument would probably be that those things are involved somehow in the recognition of that viral infection DNA. And so maybe the helicase is required, it does something that looks at DNA somehow and recognizes it as foreign and then carries out its activity. Endonuclease is a little harder to understand. They made a bunch of deletions so they could delete any one of these open reading frames, and the phage protection activity would go back to 0. And so all of these open reading frames are essential for defense. But again, they don't really know how they work. The recognition of foreign DNA or foreign nucleic acid is absolutely the key, right? Just like restriction enzymes, the worst case scenario is that they're not excruciatingly specific. Because what if they recognize their own host cells as foreign? You can be 99.99% accurate and commit suicide. That's not what you need. You need to be reliably consistent. And how that works, nobody knows. Here's another one, Thoris. And so it's just these open reading frames. This B shows up in multiple copies sometimes. Here's two A's and 3 B's, 1A and 3 B's, etc. Tightly associated with restriction modification systems, Breps and transposases and recombinases, etc. I don't think they have a clue how this works or might work. These things have an NAD binding domain. Who knows, right? Here's another one that they call Logjet. It's got 4 open reading frames. This one's different. This one doesn't provide any protection against viruses. But it does protect against transformation by plasmids. And so plasmids move around amongst bacterial strains just like viruses do, right? They don't cause cell death, but some of them may have things in them that aren't wanted. Very often, plasmids compete against each other. Infection or the presence of one plasmid prevents the acquisition of another plasmid. This is called plasmid incompatibility. And so this might have come in on one plasmid that prevents superinfection, so to speak, by another plasmid. And this is in the gram-positive bacteria. I think this one's got this one's different. Plasmids in gram-positive bacteria typically transfer as single-strand form. And so here they're looking for single-strand DNA. And so there's a DNA binding subunit. There's an ATPase. But how this thing works is entirely unclear. And so they can delete any of these and this infection goes the normal route, but when they're all there, they get transformation efficiency. It's not great. This one only provides 10-fold reduction in transformation. That's not so bad, but it's something. This is their table. It goes on a little bit more than that. So they've got each one of these systems, the open reading frame collections, the PFAMs for each of the open reading frames on here, some of the domains that are in those proteins, helicases, nucleases, ATPases, but they don't know how they work, right? Number of instances. Some of these are quite common. This is found in 2,100 essentially microbial genomes, but that's out of tens of thousands of genomes. Only 4% of bacteria have this one in it. And that's true for all these. See, most of them don't occur that frequently in microbial genomes, and they're dispersed widely. You know, some E. coli strains have one, some have another, some don't have any, some have multiple. They really are quite mobile. And some of them are membrane associated. This makes a lot of sense because viruses have to get through the membrane somehow. Topoisomerase, that's an interesting one. And so this is just showing you where they're found. Some of them are found in different kinds of organisms. So here's a pie graph of all the genomes in their database. And about half of them are proteobacteria, a bit less than half are firmicutes, bacillus, and relatives, and there's a bunch of them here that are actinobacteria, etc. And so this is kind of just the look at the whole population of genomes they're looking at. And so this one, I don't know how to pronounce these, is more frequently found in proteobacteria than you would expect at random. Some of them are more frequently found in actinobacteria. This one is almost predominantly proteobacterial. This one is a little more in firmicutes than others. Firmicutes of proteobacteria are the ones that they identify most common. Probably because we understand their genetics more, they're more frequently, they're a larger component of the original data set that was searched through. It's also true that these organisms tend to have more organized genomes than other kinds of organisms. And their whole search is based on the premise that these genes are in clusters with each other and with other genes in host defense. And so organized genomes are going to favor the identification of them. Most of these are found both in bacteria and in archaea. I found this really interesting. Archaeal defense systems, you wouldn't really know what to expect with archaea, right? That maybe they would be found, because archaea and bacteria are not specifically related. CRISPR is found in both. Restriction enzymes are found in both. These seem to be as well. Which to me sets off a really interesting question. What about protists? What about things like Giardia or Leishmania or all of these deep and unusual protozoans? Could they also have these kinds of defense systems? And I would like to do the same analysis just looking at archaea. Because if it's just found in archaea, you'll never find them with this data set. The problem with archaea is that there aren't that many genomes available, and so the data set you start with is much, much smaller. All right, so why care? Who cares? These are phage resistances. What's the point in putting this much effort into looking for them? Why should you care? The first bacterial resistance, or phage infection resistance genes that were known were restriction enzymes. Everything we do in biotechnology is fundamentally based on restriction enzymes. That's where biotech comes from. RNAi. Take a look at all the clinical trials going on right now for gene therapy. Most of them are RNAi. Antibodies is how multicellular metazoans defend themselves against infection. Can you imagine doing science these days without antibodies? You're all the time labeling proteins and things with antibodies. CRISPR. It's hard to overestimate how important CRISPR looks these days in terms of what we can do with it. And will probably be, like I say here, the wave of gene therapy in the future. And so here you have 11 new of these things to try to figure out and try to use as tools. And clues that there are many, many more, right? Let me go back. Of these 36 candidate systems, they only looked at 22. What about those other ones? Some of them they looked at but couldn't confirm because none of the viruses had any phenotype. None of the virus infections seemed to be fought off. But they could only try a handful of viruses, right? And then they thought, well, let's try plasmids too. What about other things? What about DNA that the organisms absorb directly from the environment, which many organisms do? And so there's a lot of these candidate systems that may still be very useful and may be true. And what about the ones they missed? I think there's a lot of potential in this still. So, let's go to the prompt for today. Your job, either in groups here in class or at home later on, is to think about their approach and describe in just a few sentences, in kind of a summary form, what was their method or approach in this paper. That's your primary comment. Let's see if anyone has already done one. Yeah. So, summarize the approach, post it here, and then come back later and go through people's comments about the approach and provide either feedback, things you liked or didn't like, that hadn't thought of, whatever, or improve your own and include it here. I've got here the original article. I've got the online supplement. Many of the figures I showed you come from this online supplement. There's some tables which are not very useful. And then these are the slides that I just showed you. So you've got all these resources to go with. I think this is a great approach. And I think there's a huge potential for these genes. I think you'll find that many labs in the future grab onto these things and are going to be pursuing them very hard. You can bet they patented all this, by the way. All right, go to it.